Thank you very much. Hello, everyone. So I will talk about some practical consideration in dose escalation trials oncology with investigating multiple drugs and multiple schedules. Okay, so I would like to start with some acknowledgements to Pavel and Thomas Diaki and my manager Anya. So I will mention some trials in which we have consulted Pavel and Thomas Diaki over the years. Okay, so we in this event we have heard different perspective, clinician, patient perspective, and statistician. And I would say within statistician profession there are different perspectives within statistical roles based on the role. For example, methodology expert, a consultant, someone contact research organization statistician, and I will give some sponsor statistician perspective in which the person is a member of the project team, so contributing to the drug development project, and be responsible from start point of view the trial protocol, and lastly a member of the safety monitoring committee or SRC. So I think this is very crucial in which statistician can interact with the treating investigator and can have an opportunity to listen to them. And it's not only just DLTs, right? In the dose escalation trials, there are other AEs which are not DLTs, but also important. Okay, so as background, you have heard a lot nicer presentation, Professor Christina Yap or other presenters. So this is phase 1 dose escalation trial in oncology. And here in the different model assisted design or model-based designs, there is one common underlying assumption, which is the so-called monotonicity assumption. And it is simply, we have several doses. The starting dose is informed from preclinical information, right? And then we have some planned doses, and we assume toxicity or DLT probability stays same or increases with increasing dose levels. And for efficacy, it might not be very reasonable in all cases, but toxicity, we are very confident with this. So what happens when we have multiple drugs, right? So when we have multiple drugs investigated, and consider example, you have two experimental drugs. The first in human trials, monotherapy trials are already done. Now you want to do a combination. Or another example, you have an experimental drug and you want to use as an add-on to a standard of care. So in this example, we end up with a more than a single dimension problem. So here we can consider the second one, an experimental drug. We have several doses and we have several schedules of this experimental drug. And we have standard of care, which is fixed, right? So it doesn't add another dimension in this problem. And then we have the probability of toxicity. So here, in this case, instead of target toxicity range, we have interval, we have target toxicity range. And additionally, we need to consider potential interaction between drugs. So this adds additional complexity to the question. So multiple treatment schedule, I mentioned, it might be too reasonable to consider a schedule like 1 week on, 1 week off versus 2 week on, 2 weeks off, or daily, weekly, etc. You can consider cumulative doses to order it, but it's not always reasonable for all molecules. So then the question becomes, with the monotonicity assumption I described, how can we order the toxicity properties with different dose levels and schedules in this multi-dimensional problem? So to understand this better, let's consider a toy example. So here we have different two dose levels and 2 regimens, and assume that they are they are ordered. So one is, let's say, 1 day within the DLT period, and the other is 2 days. So the DLT, the toxicity assumption we can easily make, given the regimen, we can say C smaller than D. This is easy. Or given the dose level, we can order ADC. And this is true for BD as well. But if we look at all the combinations, which is 4 in this case, we end up with 2 different toxicity orderings, C, DAB, and C, ADB. So this is the challenge, and we want to have a model which takes into account both orderings, not just assuming one. So this was a toy example. So here I'm not able to present all the details because of confidentiality, but this is from one of our motivating trial in which we have one experimental drug and we are adding on a standard of care. And here we have three different dose levels are going to be investigated of the experimental drug, and 3 different schedules. So in this case, it was duration. So duration of the administration. So it is also ordered. So, and the partner drug is fixed throughout the trial. So we start, let's say, at the first combination, starting combination, and no DLT from 4 patients observed. And at this point, safety monitoring committee, considering all different aspects, not just DLT, but one crucial part is DLT, then decided to escalate. And in the next, and at the end, we have 1 DLT from 5 patients, for example. So what methods we can use? So I would say this is like in general philosophy, it's better to start from the question, not from the method, than to try to fit the trial to our preferred method, right? So this is in line with this estimate thinking. And I don't mean to use estimate wording in the protocol, but thinking about the definition of DLT, the DLT treatment period, the time is an important component or not, or patient population, and so on. So that's why we have, it was a lot of mentioned, the communication with clinician is critical, but also, of course, pharmacometrician is very critical with this early stage, and the research experts who have done preclinical experiments on the molecule, which helped us to find the starting dose. I think these are also very critical. And with this kind of problems, we have used in different trials partial ordered continued reassessment methods, one method, and another dual agent Bayesian logistic regression model. So the monotherapy cases you have seen a lot during 2 days. So continued reassessment method, and so I'm not going to detail on this, but this is this was published in Statistics in Medicine based on one of our trial design, and in my experience so far, it's very robust in terms of operating characteristics. You have seen different metrics used, the probability of selecting correct combination, or probability of not recruiting in 2 toxic combinations, right? This kind of critical metrics is very robust approach, but of course, as I didn't mention, so this requires pre-specification of potential toxicity orderings. In the toy example I showed you, it was easy, just 2, it's done. But of course, when you have many more combinations, then it's not very reasonable to include all possible combinations, but restrict to a more reasonable subset in which clinical input very, very critical. Another one, which I will mention in the next slides, an extension of BLRM. So the methodology already published in this book chapter. So in this case, we have for interactions, we can model interactions specifically. And So if you have some prior information you want to use, you can put there. And of course, then here, the disadvantage number of parameters increased by the dimension of the problem. If you have two agents and then one agent also schedule, it's end up with a lot of parameters. Yes, so for the second one, so let's consider if the motivating trial I mentioned, and these are some specifications we have. So one drug, as I mentioned, includes both dose and schedule, and we are very interested in to be able to vary both of them. And another specification, at 0 milligram, 0 day, no duration of drug A, we would like to have that the model reduces the DLT probability of drug B alone, or the other way around. But drug B we are not changing, so I'm not mentioning. So we would like to have this kind of behavior in the model. And also we have so from previous information we are expecting positive interaction between two drugs. So, and lastly, this is the case for any, I guess, this kind of Bayesian model approach. We would like to have an easily interpretable parameters so that it is possible to communicate to clinician and to talk to clinical team to extract some prior information. So some mathematical formulas here in this slide. So this is a modified dual agent BLRM which satisfies the specification that I described. And it starts with drug A component, the experimental one. And here, this is like BLRM model, but we have also another parameter which is a like schedule slope. So we have the dose slope and schedule slope. Schedule also a covariance. So this is possible in this case because, as I mentioned, it's a duration. So in a more complicated schedules, it would have been different. But here, it's based on the number of duration, 1, 2, 3, we can put. And so using some weakly informative priors for the parameters beta and gamma, and the drug B component. So if we would have been varying the doses of drug B, then we would have another this kind of equation here, but we don't. But we have a phase 1 dose escalation trial, completed trial on drug B, and say with the same DLT, very same DLT definition. So we can use the DLT probabilities from this historical phase 1 dose escalation trial. And lastly, so this becomes the joint model without interaction. So here it's very nice because if pi A from the drug A, if this is 0, it reduces pi B, and it is same on the other way around. So as I was explaining, this is what we would like to have as a specification in the model. And lastly, so with some additional tricks, we have our interaction parameter here. Yes, so if we fit this to the illustrative data that I described, so here this is I showed you this plot, 3D plot. So this corresponds to starting combination, right? And we have the targeted toxicity region from 20% to 35%. And here we have the dose levels that we are planned to foreseen dose levels and foreseen durations. And yeah, so here you see median DLT probabilities. And even because it's a parametric approach, even untested those combination levels, we can have the median DLT probability estimate. And of course, this doesn't take into account the uncertainty, and we can have this so-called overdosing probabilities. So probability of DLT probability above the upper bound of the targeted toxicity range. So this is another criteria we use typically, and so it's desirable to have lower. And for this common threshold, 25 or 30%. So you see here we can see which kind of those schedule combinations are allowed by the model, taking into account DLT data and this model. Yes, so the simulation, about simulations, I'm not gonna show you today the simulation results. So, but I mean, in general, this is very critical, especially this kind of when it becomes very complicated. And Christina, for example, very nicely described this kind of decision tree visualizations. And it's not just communicating to clinical team, but I think for us also, it's very important. It's becoming more and more black box. So to see how it behaves, it is very critical in this kind of setting. And so I want to mention to this, so simulations are of course very relevant to regulators, and very recently a Japanese regulatory agency published a white paper, a guidance document on statistical considerations when planning phase 1 clinical trials in oncology. And so we have also experience to submitting PMDA. This is a short document, but I think very useful. And also they are mentioning combination therapy. So what they in the simulations, and they say it's needed to fully consider necessary scenarios based on the order of the combinations, and to explain the impact on operating characteristics based on simulations. And this is also one of our experience to consider scenarios in which it's not part of our subset of the ordering, potential orderings. They ask this kind of scenarios. But as I mentioned, POCRM was also gave very robust results. Yes, so this is my last slide. I have presented some industry statistician perspective in this dose escalation setting involving multiple drugs and multiple schedules, and the partial order CRM, one approach, and this I have briefly described a modified dual agent BLRM, and modification came from this actually drug B component where we use historical data. And yeah, this was repeated over and over, but I think this is also my experience, especially with this kind of complicated designs in which we would 100% needed clinical input. Close collaboration within clinical team is very, very essential. And some disclosures here and some references, and thank you all for your attention.